This video is going to be about DNA, and because I'm not really sure how specific you all got about DNA when you did the macromolecules with Dr. Lin, I'm just going to go from the beginning. And so if there's stuff that is familiar to you, feel free to fast forward as necessary. So DNA stands for deoxyribonucleic acid. It is a nucleic acid, and it is the nucleic acid that is going to be our focus when we look at how proteins are made in the cell. It's also how cells convey genetic information from one generation to the next, is in this DNA molecule. So it's a long thread-like molecule. We ourselves have 46 DNA molecules. We call them chromosomes at certain points, and they are in the nucleus. So an average human DNA molecule is about 2 inches long. And I'm sure that you already learned that DNA are polymers made of nucleotides. So we're going to specifically look at the nucleotide structure and then see how they join together to make this molecule. Each nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. When we're talking about DNA, that sugar is deoxyribose sugar. So this is a close-up of what one nucleotide looks like, and of course we're talking about millions of nucleotides joined together to make all of our molecules of DNA. A phosphate group is just one phosphorus with 4 oxygens and some hydrogens, and then there's that deoxyribose sugar. It usually looks like a pentagon, and then a nitrogenous base. Now DNA has 4 different nitrogenous bases, so a lot of times you'll just refer to them as a base or one of their 4 letters, which are going to be A, C, T, and G. So in the DNA structure, there are 4 nitrogenous bases, but they are paired up. And so as they are in a pair, we tend to look at how the shape of the pair is. And so they're broken up into 2 categories based on shape. So adenine and guanine are the same type of shape, and so they're called purines. But they pair with the other type of shape, which would be cytosine or thymine, and those are called pyrimidines. So they don't pair with the same shape as them. They pair with the opposite shape, and they kind of fit together like a puzzle piece. But the 2 shapes are called purines and pyrimidines. Purines have a double ring, whereas pyrimidines only have 1 ring. But like I said before, we're usually going to just shorten all these words up to using the letter of the base, A, C, T, and G. Oh, let me go back just a second. Okay, sorry, it's getting my arrows mixed up. Okay, so we're going to get to how they pair up in just a second, but the DNA shape is called a double helix. It resembles a spiral staircase, except it has kind of a ladder rungs across the middle, if you want to think about it that way. And so each strand on the side is alternating phosphate groups and deoxyribose. Sometimes we call this a sugar phosphate backbone. Sometimes it's just called a strand. A lot of times on pictures it will just look like kind of a ribbon, but you have to remember that they are sugars and phosphates alternating. Then in the middle is where we see these nitrogen bases, and they are going to be in pairs. And they basically make up what we consider the rungs of the ladder. So they pair based on the base pairing rule. Adenine pairs with thymine, and cytosine pairs with guanine. If you want to remember apples in the tree and cars in the garage, that's one way to remember it. But I always just tell it to myself over and over, A, T, C, G. And so that's how I remember it. But they always base pair. And so one strand determines the base pair or the base sequencing of the complementary strand or the other side. And this is very important when we looked at the process of DNA replication. This is how DNA is able to replicate and conserve its sequence. So in a molecule of DNA, we have certain segments that are called genes. And these segments code for the making, synthesis, of a certain protein. So we're going to get into, I'll have a separate video about protein synthesis. That just is a fancy way of saying making a protein. Well, it's just a section of DNA that has the instructions for that making of a protein. And there's lots of DNA that is considered junk DNA and doesn't actually code for anything. If we are talking about all of the DNA of a person, we only about 98%, or no, I shouldn't say only 2% is actually DNA that codes for genes, whereas the other 98% is called non-coding, or sometimes it's even called junk DNA. Humans have about 20,000 genes, and if we look at all the genes of one person or of one organism, it doesn't have to be a person, we call that their genome. The other DNA does have a function. It is plays a role in the chromosome structure, which you'll see during your cell division lab, and it also helps to regulate the gene activity. That means which genes are in the production of proteins in that moment and which are not. Okay, so we need to differentiate between DNA, chromatin, chromosomes. It's very important that we understand the difference between each of those words and when we're going to see each structure. So DNA is at the molecular level. That's what we're going to zoom in and be able to see when we're looking very close up. Obviously, we're not going to be able to see that with the microscopes that we have in the lab. We're talking about a size of 2 nanometers. But then that molecule wraps around some proteins called histones. And when it wraps around, it kind of coils up. You could think about threads or rope, however you want to think about it. It coils up and wraps around these proteins, and then it makes chromatin. And so that is usually the word we're going to use unless we are talking about during cell division. So that would be chromatin. And when we look at a cell under the microscope, if the nucleus just looks dark, a dark dot, if you will, or just like a dark kind of circle, then the DNA is in the form of chromatin at that moment. Chromatin is what it stays in during interphase, and it doesn't really condense into chromosomes unless cell division is about to occur. When the cell is preparing to divide, the DNA is going to make a copy of all of its DNA, and there's a separate video for that on DNA replication. The DNA will be organized into chromosomes at that point, and each side of the chromosome is called a chromatid. And so each side of the chromosome at that point is going to have identical DNA. We would be able to see this with a light microscope just during cell division. Chromatids are joined at a centromere, which is in the middle. That line is kind of weird how it's pointed. It is a bracket. It's basically the indented part. If you think of the chromosome as kind of an X shape, it's kind of that crossing point, holding those two chromatids together.